So now that we've covered all the biomolecules, we're going to start looking at metabolism, one of my favorite topics. Basically, metabolism is the overall process through which living systems or organisms acquire and use free energy to carry out various cellular functions. Metabolism can be divided into two parts: catabolic pathways, anabolic pathways. Catabolic pathways are degradative. Okay. So this is the breakdown or degradation of nutrients to generate energy. These reactions are also termed exergonic. So it's a release of energy. So we'll be looking at the metabolism of the carbohydrates, lipids, and proteins that we have learned the structures and some of the functions for. The other part of metabolism, or the other part of this pathway, are the anabolic pathways. These are the biosynthetic pathways, synthesis of biomolecules from simpler precursor molecules. Biosynthesis uses energy. Catabolism generates ATP energy. So the ATP that's generated by our catabolic pathways is going to allow the biosynthesis of required molecules. These pathways are interconnected and very tightly controlled. The other way we can describe these two pathways and their interconnection are by the coupling of oxidation and reduction reactions. Those pathways, the anabolic pathways that use energy, can be characterized as endergonic. So they absorb energy from the system in order to carry out those synthesis reactions. So this figure is actually a simpler representation of some of the metabolic pathways that converge or connect to glycolysis. Figure 17.2 in your textbook is far more complex than this, and you should take a look at it just to see that or view that complexity. But what we see here is glycolysis in the center, down this midline, which feeds into the citric acid cycle. We can see interconnections with the urea cycle, fatty acid metabolism, just to name a few. We also see how sucrose and cellulose, amylose, and glycogen are able to enter into the catabolic glycolysis cycle. Also, you'll notice that there are biosynthetic pathways that are fed, or not fed, but receive energy from glycolysis. We'll take a look at the pentose phosphate pathway, for example. So glycolysis is really central to energy production. So most cells, or all cells, have the same basic pathways, but they can be characterized by their sources of energy, sources of carbon, and uses of oxygen. And here are listed just a few of those categories. Autotrophs, for example, can synthesize all of their cellular constituents from carbon dioxide or other inorganic compounds. Heterotrophs, on the other hand, obtain their free energy through the oxidation of organic compounds. We are heterotrophs, so we oxidize carbohydrates, lipids, and proteins to obtain our energy and carbon skeletons. Phototrophs undergo photosynthesis, therefore they use light energy as an energy source. Chemotrophs can use both organic and inorganic substances as their sole source of energy. And with respect to oxygen use, aerobes are dependent on oxygen in their energy producing pathways, and anaerobes can exist without O2. So here is a figure that shows the interconnection or interrelationship between catabolic processes and anabolic processes. So on the left, we are looking at catabolism, or those energy-yielding nutrients, carbohydrates, fats, proteins, and we're going to look at the metabolism, both catabolism and anabolism of these major classes of biomolecules. These molecules are going to go under oxidative reactions. Again, here's that word, exergonic. We are producing energy, and that energy is released in the form of ATP. This is an electron carrier molecule, NADPH, and that is going to shuttle electrons into the electron transport system, which is responsible for producing ATP. The byproducts of catabolism: water, CO2, and ammonia. On the other side of this figure, we have the anabolic reactions. These are reductive. So remember, oxidation and reduction are linked. Again, oh, sorry, I should have switched my color here. I have been meaning, or forgot to mention that I was going to use red to indicate processes that utilize energy, green for those that produce energy. So on this side of the figure, again, we have the reductive processes, or the biosynthesis processes. Here's that word again. These are endergonic. So the precursor molecules here are individual amino acids, carbohydrates, fatty acids, and nitrogenous bases. From these, we can construct or build proteins, polysaccharides for storage of carbohydrates, lipids, and nucleic acids. So this figure shows the possible setup of some enzyme systems, and we will be looking at examples of all of three of these for sure. So basically, the individual metabolic pathways of anabolic and catabolic consist of sequential enzyme steps. And the organization of these enzyme systems differs depending upon the process. So if we look at the representation of A, for example, we have A, we have a substrate, and then we have enzymes, and this would be the cytosol of the cell, that are spatially disconnected from one another. And what is required here is for the substrate to diffuse, substrate and subsequent products, to diffuse between these enzyme systems. An example of this is glycolysis. So end product for glycolysis is going to be pyruvate, for example. If we look at B, we can see that these enzymes are linked to one another, so to speak. So this is a discrete multi-enzyme complex. We're going to see this in the citric acid cycle. And finally, we have a membrane-bound enzymatic system. And the example that we'll look at that represents this is the electron transport system. Metabolic processes must be regulated. if not controlled, will have too much energy or too little energy, or all kinds of detrimental things could occur. So we're looking at two different ways that pathways may be regulated. So we see in A, we have enzyme 1 going down to enzyme 5, which produces a product. And we have intermediate products along the way. The anabolic mode for this pathway utilizes an entirely different set of enzymes. Okay. So these two linked or opposing processes are regulated by independent routes. Okay. If the catabolic mode is on, that means the anabolic mode is off, and vice versa here. If you look at the B setup for regulation, I'm sorry, maybe I should do this. You notice that if we look at the catabolic mode, we have 5 enzymes. And the anabolic mode, we also have 5 enzymes. But in step 3, we have this 6th enzyme. And this enzyme can either inhibit the anabolic pathway or turn it on. And we will see this type of regulation at 2 different points in gluconeogenesis, or the synthesis of glycogen molecules. Glucose utilizes 10 enzymes, or glycolysis, I should say, utilizes 10 enzymes to convert glucose to pyruvate. But when we go in the opposite direction, pyruvate to glucose, 7 of the glycolytic enzymes are common, but 4 enzymes are going to be specific to glucose synthesis or gluconeogenesis. So this figure represents the 3 stages of catabolism. We start with large biomolecules, proteins, polysaccharides, lipids. These are consumed in our diets, and they undergo some initial breakdown. For example, proteins are catabolized to amino acids, polysaccharides are broken down to glucose, lipids yield glycerol and fatty acids. And then these building blocks, depending upon what the needs of the cell are going to be fed into either converted to pyruvate or acetyl-CoA, and then fed into the citric acid cycle. As you see, amino acids can be converted to pyruvate or acetyl-CoA, and the same for lipids. So we can also use amino acids to shuttle into the citric acid cycle, again, depending upon the needs of the cell. And from the TCA cycle, we're also going to release CO2 and water from the oxidative phosphorylation that occurs in the electron transport system. The ammonia comes from the degradation of the amino acids. So the locations of these processes are compartmentalized. They do not occur in the same location in the cell. So for example, glycolysis, these enzymes, and again, remember, they are not directly linked to one another. They are located in the cytosol. So in the cytosol, glucose is going to be broken down into 2-pyruvate. And the pyruvate then have to migrate to the mitochondria in the cell. The mitochondria houses the enzymes for the citric acid cycle or TCA cycle. These enzymes are in the mitochondrial matrix. And the oxidative phosphorylation enzymes, which aren't readily seen here, are located in the inner membrane of that mitochondria. So this also contributes to the regulation of these processes. So this is just kind of a big picture. We have ATP can be formed from photosynthesis in phototropic cells or in catabolism in heterotrophic cells. We have energy requiring activities that are powered by ATP hydrolysis. So from catabolism, we get that CO2, water, ATP. That ATP is put to work by whatever needs the cell has. And once that ATP or the energy has been extracted, we find byproducts of inorganic phosphate and ADP. So with this basic understanding of metabolism, we can now look at the metabolism of our biomolecules.